All right, buddy. We has cats. This is Noel. And here my other dog's coming in. Let's get this back to regular view. Can he do a cat dance? I've never thought to try, to be honest. She's not prickly, but she doesn't like being held very much, as you can see. She kind of puts up with me, but I'm about the only one that can do this. You know. And here's Pepper being all kinds of curious. And there's kitty cat. It's the great canine feline standoff. Oh, my laundry as well. Probably didn't need to see that. Okay. She's cute. All of them are. Gonna see if she starts beating up on Pepper in a little bit. Our other cat, Caramel, likes punching Cricket in the face quite a lot. Because so far, Cricket doesn't have the sense to leave her alone. She's kind of dumb. She's a dog. All right, let's get busy. So today's when we start the material basically. And so I'm going to share the screen just yet. I want to go over what we'll be covering this section. So this grumbles and grumbles and grumbles. So as I told you last class, course is split up into 4 sections, and each section has its own exam at the end of it. So this section, section 1, is really going to be partly on recap, which is not going to take us very long, a couple of class periods, including today, most likely. That's just to get you up to speed on language and some basic concepts from Gen Bio 1 and the like, largely because some people haven't done this for a good long time. What's next? Then we have Mendelian inheritance and math, so basically probability and some stats. Not a huge amount, but whatever, what's useful for understanding Mendelian inheritance. Mendelian inheritance is really all about, yeah, I know, I'm sorry. Got a comment in the chat going, use stats, which I could wholeheartedly agree with. But it's useful, right? So that's why we're going to talk about it. So Mendelian inheritance is really all about probabilities, really. It's all about using what you know about inheritance and properties of alleles and so on and so forth to working out the probability that you get a certain genotype combination and phenotype in the offspring. Really, that's all it's all about. So that's why we need the probability and stats. The stats is really all about seeing whether we get what we expect, right? So that's the kind of stats that we'll be doing for that. And that'll also tie into the lab as well. We'll be using that later on. All right, then, but to be honest, Mendelian inheritance is kind of fun in the sense of, you can use it to kind of like work out little logic puzzles and stuff. But really, it doesn't capture even a tiny fraction of human genetics, right? So most of the reason the way you are, right? Be it, you know, physical attributes, hair color, eye color, you know, height, you know, all kinds of things like that. And even kind of more complex stuff like behavior, right? And, you know, the diseases that one accumulates over the course of our lives, right? Those are all non-Mendelian. Multi-gene, leave her alone. Multi-gene traits that have, hey, get out of there. Kind of chasing the cat. That have alleles with different properties, you know, that don't follow the usual dominant recessive kind of format that we're familiar with in Mendelian inheritance, right? So almost everything, bar simple single gene congenital diseases like cystic fibrosis, sickle cell disease, phenylketonuria, you know, there are plenty of others, right, which I couldn't recall off the top of my head. But other than those, everything else is multi-gene, right? Get out of there. I don't know why, but the cats love eating cat poo out of the litter box, which I think is just disgusting. So try and keep them out of the bathroom, because that's where the litter box is. All right, so a big chunk of what's left of the section is non-Mendelian inheritance. That's the really fun stuff when it comes to talking about inheritance. Then to finish up, we're going to do DNA replication, cell cycle, and cell division stuff. Because that also ties into, put that down a little bit there. There we go. That also ties into all of inheritance, right? So this is essentially what's going on at the gamete level, I guess one could say, the cellular level. This is what's happening at the level of the individual. Right. Okay. So really what's next is just recapping basic knowledge, really. So some of you, this will be kind of a recap, old news, essentially. Some of you, it might be something that you learned many, many moons ago and need a kind of bit of refreshing. Right, so where's my, here we go. Oh, sorry. Busy day. So yesterday I learned how to use the annotate function, which is kind of fun. So I'll be tormenting you with that today. It's kind of fun. I like using it. It's super, super tedious in a sense, right? Because I'm going to have to write stuff. Oh, hang on. Hey, maybe I don't. All right, well, that would certainly save a lot of long-winded finger drawing. Okay, so we're going to start with this. I like starting with this because there's a huge amount of information in this diagram, which is super relevant to a lot of biology, a lot of genetics, right? And so obviously this is one of two strands of a DNA molecule, right? Where's my chat? Chat. I'll type up stuff as I go along. So DNA is an anti-parallel double helix, right? So that's kind of an important definition to make note of. Didn't have my morning coffee today. And we'll get into that a little bit in, I think that's like the next slide along or so. But anyway, to begin with the basics, right? So this here, hopefully you can all see this, right? This is the building block of DNA, right? So this is a nucleotide, right? It's a nucleotide because it consists of 3 parts, right? We have phosphate group, right, which is that bit at the top with the P in there. This is negatively charged, right, because of these OH groups essentially losing a proton. Actually, give me a second. I'm just going to close the door. My wife's taking calls. Some of the patients she speaks with are pretty deaf. Yeah, DNA as a whole is always negative because of these negative charges. You can see them. There's another one here. There's another one here, right? They will be all the way along the backbone, right? So that's a central property of DNA. Oh, you're welcome. It's important in terms of chromatin formation, right? So chromatin is DNA and protein. So DNA doesn't look like this, or even like, sorry, got to get off of this. That one doesn't even look like this. Oh, right. In a normal state, right? DNA is always complex protein, and part of that attraction is managed through charge, right? So its charge is super important from that. It's also really important when you want to run a gel. So if you want to separate DNA based on size, then you do so by its electrical charge, right? By applying a voltage, and essentially the DNA gets attracted to the positive end, right? So it's a really, really important property of DNA. There's also a 5-carbon sugar, ribose. That's this bit here, right? Looks like a cupcake with a cherry on top. And a nitrogenous base, right? Which is this part here. So those are the adenines, guanines, cytosines, and thymines, right? So let's get that stuff clear. So these nitrogenous bases come in 2 basic forms, right? We have a 2-ring form, right? So that is those are called purines. And that's adenine and guanine. Almost tripped up there. And single ring nitrogenous bases, those are cytosines and thymines. These are pyrimidines. Easy way to remember. Y, O, Y, If I can spell it right. Am I such a small base? That, oops, equals pyrimidines. So all of the bases with a Y in them, cytosine, thymine, are pyrimidines. Uracil as well, but that doesn't work so well. And they're small, right? Why am I such a small base? That's how I remember it anyway. Okay, so another thing that you'll notice, right, is that those nitrogenous bases have polar covalent bonds in them. There we are. Not 100% sure about that one there, but there's got to be one of them somewhere. Oh, actually, you know what? It doesn't. There you go. You'll see why in a little bit. Okay, so let's get back to drawing. Here we go. Right, so nitrogenous anybody remember what kinds of bonds, name of them, not James, forms between a slightly positive and a slightly negative region caused by a polar covalent bond? I'll type that out in the chat. Yeah, is that Muntari? Not sure if I pronounced that right or not. Hee. Second time lucky. I'm going to finish typing this out because you kind of beat me to it. Yeah, it's hydrogen bonds. Now, what sort of strength or weakness do hydrogen bonds have? If we're going to rank them between the strongest and the very weakest, kind of where in that spectrum do hydrogen bonds fit? Can anybody remember? Almost. The weakest are van der Waals forces, which are nuclear attraction. I don't actually fully understand what those are, but they are the second weakest, right? So you're all pretty much correct. They're one of the weakest forms of bonds, right? And the way that you can remember this is that hydrogen bonds are what hold water molecules together to make water, liquid water, right? And you don't have to heat water up much, and those water molecules start flying all over the place. They get separated from each other, right? So they're weak enough that you can break them apart, but strong enough that, you know, insects can walk across them, stuff like that, right? Or even things like, oh, I'm sorry, surface tension. If you ever seen videos of that astronaut on the space station wringing out a hand towel soaked with water, really cool. Basically, the water kind of like creeps along his arms in a film because there's no gravity to break that surface tension and cause the water to fall. Super neat. Okay, so hydrogen bonds have a really cool property, very useful property, in that they are weak individually, but if you have enough of them, they're relatively not strong, but capable, I guess one could say.
And it's hydrogen bonds that hold together the two strands of DNA. Let's see if I can. And they're always drawn with these vertical dashed lines, like so. Ish. Better than that, obviously. Okay, so one of the benefits with DNA of having hydrogen bonds hold those. Let's see if I can use text, because that's going to be so much faster. Come on. There you go. Hydrogen bonds, right? Is that they can hold DNA together. DNA is a very stable molecule, right? You can find it in bones of things that have died, up to around 50,000 years ago, which is kind of crazy. Everything else may have just disintegrated, but DNA inside bone marrow and things, you can still get it out, and it's more or less intact. So H bonds hold DNA together, or at least the two strands, but allow it to be separated relatively easily to allow for DNA replication and transcription. So it's a real key property of DNA. DNA, you want to stay shut until you need it, then you want to be able to unzip it and open up when you do. Okay, so another thing which you'll see here, right? It's actually kind of actually one thing that I don't understand. I think there might be another one in here somewhere. Maybe it's that one's a question mark. It kind of doesn't work otherwise. How many bonds do you think holds the, oh, actually, before we even get there, can anybody remember what bonds with what in terms of nucleotides or bases? A and T and C and G. Excellent, Alicia. And Israel, and yeah, her too. Good job. So here you can see why, right? So for A and T, or A's, we don't have a T up there, we've got two possible apples in the trees and carrots in the crown. Well, that's clever. Never heard of that one before. Makes sense. Easy to remember. Whereas C's and G's have three. And it's all down to their shape, right? As to how many, well, their shape and how many of these polar kind of charged groups stick out from the edge of them. And C has to have three because otherwise it doesn't work. So, A's and T's are 2H bonds. C's and G's are 3. And that's going to be super important later when we talk about the ease of pulling apart DNA strands, right? So if you want to separate DNA strands, then you're going to want to have lots of A's and T's because it's going to make it easier. Okay, so that's some key stuff. Oh, also, you wanted to move on, but I have to remember to need to cover certain key things. Second thing, one thing you notice that if A's and T's go together, one is a 2-ring, purine, one is a 1-ring, pyrimidine. So you always have a 2 ring with a 1 ring. Right, so you always have essentially 3 rings between each of the things. So in terms of base pairing, right, A's and T's, C's and G's, apples in the trees, carrots in the ground, which is a super cool mnemonic to remember with, is about number of H bonds, size of the bases, the whole 2 + 1 deal, if I could write that properly, and the shape of the actual bases, right, how they kind of fit together, right? And that's what allows DNA to stick together, also what allows us to copy it really faithfully, at least in part. There are other things as well. Okay, anybody got questions so far? No? All right. So something else. Both C's and G's and A's and T's are held together with hydrogen bonds, Richard. The difference is the number. A's and T's have 2, C's and G's have 3. Makes them, C's and G's basically stick together a little bit more strongly. Okay, so I really like this image for a lot of reasons, but there's a lot going on here. So another thing that is worth pointing out is the 5 and 3 prime ends. So DNA is unidirectional. It is different from one end to the other. At one end, you will have a phosphate group, which is attached to the 5 prime carbon that you see here. That's why it's called the 5 prime end. So all carbons in sugar molecules are numbered. Let's go 1, 1 here, 2, 3, 4, and 5. Right. Now the other end, we have a 3 prime hydroxyl group. So hydroxyl, in case you've forgotten, is a OH group, right, down here. And that is attached in DNA, at least, to the 3 prime carbon, or the number 3 carbon. That's why this is called the 3 prime end. And also, this is the only spot. Oh, hang on. See if I type this out. This is the only place that you can add a new base to, right? So DNA not only has a direction, but it can only be made in one direction. It can also only be read in one direction too. The other way around, Alicia. So the phosphate group is attached to the 5 prime carbon. Oops, if I can spell it right. Phosphate is a real hard thing to type. Oh, okay, good. Basically, it's the number 5 carbon. And the OF. My typing sucks. Not quite as bad as my writing. Yeah, I was already typing that, figured I might as well continue. But yeah, exactly what you said. The OH is always attached to the 3 prime carbon. And this is what's used to create this phosphodiester bond, particularly this one here. Right? So you can see why you have to have that OH, right? So this is essentially an unused one. This is a used one. And when you do add a new base, this is the phosphodiester bond is what is produced, right? So this is what holds together the backbone, right? So the backbone of DNA is alternating sugars and phosphate groups, right? So sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down till you get to the end. Or if it's bacteria, you never get to the end because it's circular. Okay, so that's probably enough from that one. Let's get rid of those. And this directionality is super important in terms of DNA replication and transcription, perhaps even more so in DNA replication, because it causes all kinds of issues. Oh, sorry, I'm not bored, I'm just tired. I haven't, like I said, I foolishly skipped my morning coffee because I was busy doing something else. All right, so not much extra to see here. The only thing that I want to highlight is the fact that DNA is not symmetrically twisted, right? So, a couple of things actually, before I forget. So there is what's called the minor groove, right? Which is here, that one. And there's the, yeah, that didn't work out so well. I was so excited not to have to finger write everything because that was just painful yesterday. And then you have the major groove. Oh, well, maybe that doesn't work quite the way I thought. And then you have the major groove. You also have this anti-parallel business, right? So if you look at this carefully, this strand that goes all the way up, oops, this is really hard to do. Because my finger slips and then it's gonna like, fine. Gonna start getting mad. Go the other way now. All right, so we got, see if I can get it done before my finger slips again. There you go, that'll do, more or less. I only got a small trackpad. You'll see that one strand goes down here. The other strand goes in the opposite direction, goes up, right? So if you were to be pedantic about this, really all the letters in this strand should be upside down, right? Because it's essentially upside down, going upwards. All right, so we're not going to talk about it today, but this DNA is complex into much more complicated structures, larger scale structures called chromatin, right? We'll talk about that later on. But what I want to do now is quickly go over some of the terminology and like for gene expression, right? So really, the minor group, the first part of the DNA. Oh, no, what I was trying to point out is, see how the when you twist the DNA, right? So it's a spiral. It's not like twisting together two bits of wire or pieces of string. It's not an equal twist, right? Yeah, exactly. So you have the tighter twist in a sense, and then the more open twist. It's like a cack-handed attempt to twisting a pretzel, for example, right? You don't get it even. This one's closer together. This one's larger apart, right? So you don't have an equal twist. Yeah. And this is really important in many cases for proteins that recognize DNA, because proteins will actually recognize, say, the major and major groove, right? They don't bind discriminately. They actually have to fit a shape of the DNA, right? So moving on to gene expression. Gene expression, if I spell it right, it's basically DNA gets copied into mRNA, and that's used to make protein, right? So proteins are the things that do stuff. Also, non-coding RNAs as well, but we'll get into those another time, right? So also, in eukaryotes at least, I'm just going to call them eukes because it's easier to type. There's the issue of location. DNA is in the nucleus. Proteins get made in the cytoplasm. So mRNA is an intermediate in a true sense, right, between DNA and protein, but it's also an intermediate in the sense that it allows information to be carried from one place to another. Right. So how that works in terms of generating mRNA, getting it to the right place, translating into protein, all of that is underneath the kind of the big umbrella of gene expression. And it's arguably one of the most important processes in biology, right? Because it really is a large part of why you are the way you are, right? And so that's kind of put in this kind of neat little flow chart, right? So DNA here, right, doesn't do anything, doesn't go anywhere. It is a fairly dynamic molecule, but it's purely a storage of information, right, and a mechanism for inheritance. It does not do anything in and of itself, right? That is done by either RNAs or protein, right? And so the flow of information is really from DNA to mRNA through the process of transcription, and then using these non-coding RNAs, ribosomal RNA and transfer RNA, plus the protein scaffold, which is the ribosome, you then generate protein through the process of translation, right? So that is, well, I mean, it's kind of easy to say, there's an awful lot to it, but that's gene expression. Quite quickly, I want to go back up, go clear those off. I want to jump ahead just a little bit. So before we get into expression, just want to talk a little bit about RNA, because RNA is actually pretty cool. If one were to have which is the most interesting nucleic acid competition, which one may, I guess, RNA would win it hands down, because it is easily the most interesting, one of the most interesting molecules. Proteins kind of have it licked in terms of utter complexity, right? Because they're much larger, more complex molecules. But RNAs are actually pretty cool. And so RNAs are actually, they're the only molecule that can store both information and act as an enzyme. And the fancy word for that is a ribosome. That's an RNA enzyme, essentially. Okay. And so, cool, kind of diversion, distraction. That's actually the basis of one of the theories of how life originated on Earth, right? It's called the RNA world hypothesis. And so life had to originate somehow, right? We didn't just blink into existence, at least in terms of scientific theory. And molecules that are able to both store information and transmit that information from one molecule to another, like in sense inheritance, right, are a pretty compelling beginning, right, to that process. And RNAs, and there are a few other types of nucleic acids, TNAs and PNAs and GNAs and things like that. Because certainly, even though we only have RNA and DNA, those are not the only types of nucleic acids you can have. But RNA is a pretty compelling beginning to life on Earth. And they have all kinds of like funky structures. So RNAs can bond to themselves, right? It's what you see here in these stems, right? So you have double-stranded RNA here and here, right? You can have these loops, right? And you can also have catalytic domains as well, right? Which are the parts of the RNA molecule that actually do stuff. So good example of a ribosome is ribosomal RNA, if I can spell it. And ribosomal RNA forms the peptide bond between amino acids. Right, it's not the ribosome. It's not the, well, I mean, the ribosome does, but it's not the protein in the ribosome. RNAs are also incredibly fragile too, which is both a good thing in terms of control of gene expression and a bad thing if you want to actually study them, because they disappear in a puff of smoke, basically. Well, not literally smoke, but just look at them funny and they're gone. They're very fragile molecules. That's why when you get ancient molecules, they're never RNA, they're always DNA, right? So brief overview, right, because we only got 6 minutes left, of, actually, let's clear this off. Translation is, it's the best way of putting it. It's the reading of mRNA, which consists of 4 letters, but it is read in groups of 3 letters. Those groups are called codons. And that's how you can go from 4 bits of information, and that's in the kind of computer code kind of term bit, right? So it's A, C's, T's, and G's. That's it, the nucleic acid, or A, C's, U's, and G's, if we're talking about RNA, right? To 20 different amino acids, right? Obviously, if you only had one amino acid per base, you'd only ever be able to have 4 amino acids, right? We've got 5 times that many. However, if you do the math, which is, I always get around, so I'm not going to tell you the wrong way. Get it the wrong way around, father. Anyway, if you do the math, you end up with 64 different possible words, right? From 4 letters. Actually, it's kind of funny if you think about the human language, or at least British or Spanish or the other Latinate languages, got 26 letters. You can get many words you can get from that. All right. So anyway, those different words make up what's called the genetic code, right? And so the genetic code is called degenerate because typically more than one word says the same thing, right? So, for example, there are 6 different ways of saying the word Lucy in terms of translation, right? Each one of these codons all can code for Lucy, right? So the exceptions to that are methionine. And so this is what's called the start codon. So it's at the beginning of every single protein produced, whether it's prokaryotic or eukaryotic, viral, whatever. Oh, heck no, yeah, that is definitely not on the cards. Well, let me characterize this. All of this, like the whole table, no. Certain things, yes, right? So if you ever need it, which sometimes you do, I provide the chart for you, right? So I don't expect, I can't remember what this is. I mean, I can remember a few of them, maybe. Maybe. I can remember all the stop codons and the start codon and maybe the odd other ones. I can always remember AAA as lysine because that's like roadside assistance. But also, in the little bit of time we've got left, there are 3, I can circle these and no others, stop codons. So whenever the ribosome comes across one of those, transcription stops. Or not transcription, think back, translation stops, right? So these are really important for stopping it where it's supposed to be stopped. But also if one of these is generated through mutation, this is tragic and catastrophic. And it always results in the loss of function, right? Every other amino acid, with the exception of tryptophan, which we'll come across when we talk about bacterial gene regulation, every other one is encoded by more than one codon, right? So this is what's meant by the degenerate genetic code. More than one codon equals the same amino acid. And this is a big boon in resistance to effect of mutations. So if you look at, let's clear the drawings. Here's a good example, serine. So if you look at serine, as long as you have UC at the beginning, the first 2 letters of the codon, the last one doesn't matter. Just going to finish this up and then we'll be done. The third one doesn't count. And that's called wobble. Basically, it means the last one, I don't know, the tRNA doesn't care about it so much. And so if you have one of these bases changed, it's a mutation, but it doesn't change the amino acid. Now, if you went instead, by random chance, had the first letter, the U, changed, well, now you've got proline or threonine or alanine, depending on what that letter was changed to, right? So mutations in the third letter or position of a codon don't typically matter. Sometimes they do, like you could go from histidine to I felt like saying Guatemala for some reason. Glutamine, don't know why that popped in my head. You can change, but it's like a buffer against mutation. All right, we'll finish up the last few slides or equivalent of in class next week. So next week is in person. Woohoo! And that's it for me. So let's clear these off. Thanks for coming, everybody. Great to have you again. Look forward to meeting you all on. I'll answer you in a bit, second, Val. See you all on Monday. So, Val, the only different characteristics the minor and major groove have is what can actually fit inside them. So it's just a physical space characteristic, right? So the bases that make up the minor and major groove, I mean, it doesn't, which bases are there doesn't cause that. It's somehow the way that the DNA twists around itself causes that major and minor groove. All right. Thanks very much, everybody. Catch you next week. Let's stop share. And time to go.